Hi guys, I thought I'd take the opportunity to put together our next podcast. Obviously we looked at some class end of last week and start this week, but for those who want to review it, here we go. The Chemical Nature of Cells, Chapter 1. So this chapter of course covers a lot of stuff that's in our first SAC, so probably worth just reviewing it carefully and making sure you've covered all this detail in your SAC preparation, particularly in your background. So we have three reasonably important characters, Theodor Schwann, Matthias Schleiden and Rudolf Virchow, who proposed, based on the work of hundreds of other biologists, that all organisms are composed of cells, all cells come from pre-existing cells, and the cell is the smallest living organisational unit. Sorry about that, I just scratched my nose. So this is very much like chemistry, isn't it? Where the atom is the smallest component of a element, and all matter is composed of atoms. So the same sort of process. It's a really important idea, though. It's one of those three central tenets of biology as we study it, and we'll look at the others as we go through the year. The cell, of course, is composed of a number of structures. We tend to use the word organelle, but we know that some of those things are in fact not organelles, organelles being membrane bound essentially. So we have a plasma membrane around the outside of the cell. Of course in plant cells we see the cell wall, the defining feature of a plant cell. Within that plasma membrane there is fluid. The fluid itself is called cytosol. Fluid with organelles apart from the nucleus contained within them we call cytoplasm. That's this definitional, so don't worry too much about that. The nucleus, of course, contains the DNA and controlling all the proteins and biochemistry of the cell. Mitochondria taking glucose and reducing it so to release the energy for all the cell's processes. Endoplasmic reticulum, where proteins are synthesized, smooth for internal use, rough for external use. Essentially, ribosomes, where the information held within the nucleus is translated into a protein structure, well, a primary structure. We'll talk more about that in about chapter 3. The Golgi apparatus, where proteins are finished, if you like, and packaged up ready for release. Then we have centrioles, which take a rather important role in cell division, which we'll talk about a bit later this year. Lysosomes, which contain chemicals which cause a cell to lyse and therefore often called a suicide organelle, because if they rupture and release their chemicals, then the cell will rupture and fall apart. And this is involved in a process called apoptosis, so programmed cell death. Plastids, there's a huge range of plastids found particularly in plant cells that offer storage components, all sorts of interesting things. Vacuoles, large ones of course in plants, small ones in animals to some degree, in some animal cells, and these are membrane bound again of course. Vesicles, which basically bud off the Golgi apparatus containing the substance you wish to take out of the cell. It'll go to the membrane, become part of the membrane and release its contents. And we've probably talked about endocytosis, exocytosis and pinocytosis already, haven't we? Chloroplasts, again, like the mitochondria, contains its own DNA, so we know these are once prokaryotic cells that are living in symbiosis within this structure, and they allow plants to take sunlight energy and create a store of chemical energy that we call glucose. Large vacuoles, of course, in plant cells, which fill with water and apply turgid pressure to the cell and keep its structure. The cell wall, which of course gives cells that much more geometric, regular size, shape and structure than a plant cell, and give it strength. Plants, of course, lack any skeleton. And tonoplasts, which are a particular type of plastid found within plant cells, which you don't have to worry too much about at this point in time. We talk about cells being either prokaryotic or eukaryotic. So cells go back a long way, and there would have been a time when there were thousands of different prokaryotic cells that evolved and failed, and some survived and some still survive, and bacteria are one of those groups of prokaryotes that causes some problems, and we can use them for useful things too. But we evolved these things called eukaryotes, which are like us. They're much more complex, they organise much more detail, and of course allow for this huge diversity of life we now see on the planet. Prokaryotes lack membrane-bound organelles, and their DNA and most of the rest of the cells are simply floating about the cytosol. They do of course have ribosomes, but ribosomes aren't organelles. It's one of those nice little confusing questions I'd like to throw into an exam paper. Prokaryote still needs ribosomes to be able to take its DNA and interpret it and create proteins. Well, got the generalized plant cell there. I'll skip over these. You've done this often enough, but worth having copies of somewhere, just for reference. Hopefully in your cell organelle PowerPoint, which you can use to revise. There's our animal cell. And then of course the really important part of this first chapter is that idea of all these molecules that are within cells and outside cells that are wanted by cells, and how in fact they move and cross membranes. So the important inorganic, those which don't contain carbon, hydrogen and oxygen, and from a biology point of view aren't produced by organisms, water, oxygen, carbon dioxide, nitrogen, the various mineral salts, and this idea of pH and acids and bases becomes really important, and we'll talk about that with our next act when we look at enzyme activity. So water, of course, is critically important because all life began in water. And cells all need water, so somewhere between, I think it's actually about 60 to 90%, depending on the organism, of all organisms is water. And it allows for things to move. It allows for osmosis to occur, it allows for diffusion to occur, so that materials can move into and out of cells much more effectively. Water is incredibly important and has very, very interesting properties. As we know, it actually becomes a solid when it cools, which is the only one it does. So it acts as a solvent, so it dissolves things, which allows the stuff we want to get into and out of our cells the ability to be moved. Has a very high heat capacity, so it can withstand quite a bit of heat before becoming a problem, which means it doesn't become a vapour until quite a high temperature as well. And of course the plants love the fact that it's highly cohesive with a huge degree of surface tension. So we think about the tree in the image behind me, that's my favourite gum tree in my backyard. It uses this surface tension and cohesiveness of water to draw water up from the soil, which at the moment is quite wet, and flush the whole plant with fluid and allow it to be able to do what it needs to do. So all its movement of materials and storage of glucose and all this, that sort of thing. And all this information is related back to the way it bonds and that polar molecule you can see there. So critically important stuff, water. You're not going to get examined on water's chemical structure, but the knowledge that water is critical and why it's critical is important to you. Of course, we breathe in oxygen. We need it in our mitochondria to burn the carbon dioxide and release the energy. Interestingly enough, only about 21% of what we breathe in is oxygen. So we're not using a lot of it, and we breathe a lot of it out before it gets a chance to exchange with carbon dioxide as well. But we desperately need oxygen, and one of our big homeostatic processes is making sure that our oxygen and carbon dioxide levels are under control. We, of course, produce carbon dioxide as a byproduct of having destroyed that glucose. Well, not destroy, I shouldn't say that, should I? Sorry, Mr. Lavoisier. Sorry, chemistry students. We have transformed glucose into carbon dioxide, water, and released the energy it contains. But of course, that byproduct of carbon dioxide is not much value to us. Great value to plants, so they of course also do cell respiration and break down the glucose they make in mitochondria, and need that energy in themselves, and they can reuse their carbon dioxide to make the next lot of glucose. For us, it's waste, and we've got to get rid of it. The atmosphere contains about 1% carbon dioxide, but that sadly, of course, is rising, which could cause some interesting difficulties for animals and plants. Nitrogen's most of our atmosphere. 78-79% of our atmosphere is nitrogen, and yet we can't breathe it in and use it. So we breathe it in all the time, we breathe it straight back out again. But it's critically important in the building of proteins. So getting hold of nitrogen is a really important thing for all living things. And there are many plants that find ways of doing that by having symbiotic relationships with bacteria and soil bacteria, things that allow them to take up the nitrogen and pass it on. And you would have had a look at that last year. we won't bother with it this year. Minerals, of course, we need so many different minerals. Phosphorus, potassium, calcium, magnesium, iron, sodium, iodine, sulfur, in reasonably small amounts. And the other ones on that list in even smaller amounts. But these are critically important to different proteins, different lipids, and to different structures within our body. Some of them, of course, act as what we call coenzymes. So you need vitamin D in your diet to help you take up the iron. And the iron is needed, of course, to make hemoglobin in our blood. So all these things are only required in small quantity, but critically important to all plants and animals. And we get most of it from our plant material that we eat. Hopefully you're aware of the pH scale. pH actually apparently stands for portents of hydrogen in Danish or something, which is the language of the guy who produced this system. So the more hydrogen ions you have, the more acidic a substance becomes, and the more hydroxide, OH minus ions, the more alkaline a substance becomes. And of course, if you've got a balance between hydrogen, H+, and hydroxide, OH minus, you have a neutral situation. You have water. So water in its purest form should be about 7 pH. Of course, we don't see it. We have things always dissolve into it, so it's always somewhere between 6 and 8. So it's slightly acidic to slightly alkaline. Interesting enough, hydrochloric acid is about 0 pH, and our stomach contains hydrochloric acid. We'll talk more about in our next sack, in our preparation for our next sack. Of course, there are also organic compounds, and these are the ones that contain carbon, hydrogen, and oxygen, and that we actually make. These are things we create within our bodies. So carbohydrates, essentially a huge source of energy for all living things, but they also form structural components. Cell walls, of course, in plants, but also things like the nails you've got in your fingers there and your hair, partly carbohydrate. They also form the backbones to DNA and RNA, and we use them essentially for just the energy store that they bring. We talk about carbohydrates being monosaccharides, disaccharides, polysaccharides, and oligosaccharides. The monosaccharides are things like glucose, and we obviously want glucose because it's the simplest form we can get to break it down. And most of our carbohydrate we eat is broken down to glucose because it's small and can reasonably easily move across cell membranes. And we'll talk about that sort of thing later because it's by active transport, by facilitated diffusion, sorry. We have disaccharides like sucrose, so smaller units put together. Starch is a classic polysaccharide, and chitin's one of those oligosaccharides. We'll have a look more of those a bit later. Proteins, wonderful, interesting things. You can see there the primary protein structure. We're going to talk about transcription and translation of DNA, and we'll in about chapter 4, I think. And that's simply that primary structure, simply the amino acids as the ribosome has read the information in the messenger RNA. That then moves into a endoplasmic reticulum, and they become coiled. So we see the alpha helix, and then pleated. So we get these alpha helixes and beta pleated sheets. And so we've got this secondary structure. That secondary structure then folds around itself and creates those active sites that we talk about. And we get this tertiary structure. That then leaves the endoplasmic reticulum and goes out to the Golgi apparatus for packaging for external use. At that point, you can create a quaternary structure. So a couple of tertiaries are wrapped together and create this quaternary. And the classic quaternary for us is hemoglobin. It's made up of about 4 different proteins. So it's a quite complex protein. DNA and RNA are also really important and formed from carbohydrates and proteins. And again, we'll talk much more about this because DNA becomes extremely important to us in Unit 4 when we start looking at genetics, and a bit later in this area of study when we talk about protein synthesis. But the DNA, of course, exists as very long coiled chains. They have a sugar backbone with phosphates and these proteins that are called nucleic acids which join across to form the helix. And we'll have a look at those in much more detail as the year goes on. Lipids, we used to call them fats and oils, but of course we can't use the word fat, and not politically correct, but scientists call them lipids. These are non-polar, and they're hydrophobic. They don't like water much.